morning, ma'am.
Good morning, ma'am.
Good evening, ma'am.
Good morning, ma'am.
All of you mute yourself. If you have any doubts, you can ask at the end of the session, okay? So today's topic is about the cell division. Before I start the topic, I would like to welcome all of you to our institution. Welcome all of you. I'm Dr. Sandhya Yatagiri from Department of Anatomy, Associate Professor from Department of Anatomy. This topic is slightly a revision for you, probably. Mute yourself. Slide move. Cursor. Okay. So before we start with the cell division, let us see why do the cells divide, right? We need to understand why the cells divide. See, all the cells will have the cytoplasm and the nucleus, right? And they keep growing. When they grow, the distance between the cytoplasm and the nucleus, that is very important. So the cell receives the nutrition, but as it grows, what happens is the distance between the cytoplasm and the nuclear membrane, that goes on increasing. So what happens is the cell is deficits of its nutrition. So what does it further do then? If it divides, then what will happen? It maintains its size, right? So the distance between the cell membrane as well as nuclear membrane, that is maintained, and hence the nutrition can be given to both the daughter cells equally, right? So the main purpose of the cells to divide is, one is to maintain the growth of the cells. Then, as I said, it is one of the biological necessity to provide equitable nutrition to the cell. And third is for the repair and replacement of the dead cells. So this you would have seen in a number of occasions, like the skin. That is a classical example to replace the dead cells, right? So as we see that skin will shed off the epidermis, the outermost layer, the keratin layer, that goes on shedding normally once in 28 days. There is a cell cycle there. It goes on shedding and we are replaced by newer cells. Then next reason to divide is one of the methods of asexual reproduction. Now, when we see methods of cell division, there are two methods. One is the direct, that is amitosis, and then the indirect, that is the mitosis. So here I have just put a slide to show you amitosis. Here what happens is, this is the cell with the nucleolus here, cytoplasm. It is just a direct division of the nucleus as well as the cytoplasm. So a constriction appears at the cell membrane that goes on deepening, and even the nucleus divides here. Just a simple division of the nucleus and the cytoplasm to give rise to two daughter cells. This is not seen usually in the animal cell, mammalian cells. So let us move on to mitosis. Before telling about the mitosis, you all know the cell undergoes a cell cycle, right? So in the cell cycle, what all we see? We see the interphase as well as the mitotic phase. So there are a lot of things which happen in the interphase. In the interphase, we have gap phase as well as synthetic phase, right? So as soon as the mitosis is over, the cell moves on into the interphase. In that, first is the gap 1 phase. So what happens during the interphase? We see there is synthesis of proteins there, right? Enzymes such as all these for the growth of the cell, right? So it goes on synthesizing the requirements of the cell. That is gap 1 phase. Then we have the synthetic phase. In that, mainly we see there is DNA synthesis. So there will be replication of the DNA which will occur during the synthetic phase. So here you need to remember, if the cell enters the synthetic phase, it has to divide. Okay, it has to divide. So only those cells which are destined to divide, they enter the synthetic phase. Then you have the gap 2 phase, interphase, that, okay. Then it enters the mitotic phase. So here we'll study in detail the mitotic phase. So let us understand which are the cells which undergo cell division, and there are some cells which do not divide at all after birth, right? So categories of body cells. There are three categories. Broadly, we can divide them into three categories. The first is they do not divide at all postnatally. Once the child is born, okay, the fetus is born, you would see that these cells, specialized cells are there, they never divide. Example, the nerve cells. Clear? Nerve cells, they don't divide. Then second category of cells, they continuously they divide. Clear? Because of wear and tear in the body, you need to replenish the cells there. Example, the intestinal epithelial cells, they have usually even the epithelial cells of the stomach, right? They will be having about 4 to 5 days, 7 days, like that cell cycle will be there. They go on dividing to give rise to newer cells. And as I said, epidermis of the skin, that also goes on dividing from the stem basal cells. You see that the cells go on reproducing newer cells to reach the upper layer of the epidermis. Then third category of cells are there. These are highly specialized cells. I am not sure whether you are aware of these cells, the liver cells. Okay, so usually the liver cells don't divide. The hepatocytes, they are called as hepatocytes, they don't divide. But what happens at times, there is, you think there is a some tumor, or could be the cirrhosis of liver is there. You want to remove part of the liver. Here, you want to remove part of the liver. So you see that part of liver is resected out. Then what happens? The liver takes off the function of division of the cells. That means it has the capability of undergoing mitotic division so that newer hepatocytes are produced and they replace the dead ones. So liver is a classical example of this. Not all the times liver cells divide. Clear? Only if you want to, if you have resected about 2/3 of the liver, you think. Clear? And only 1/3 is left. So that 1/3 has the capability of going on dividing and replace the dead cells. So these are specialized cells of the body. So let us start about the mitosis. Now, you know, mitosis occurs in all the somatic cells of the body and also the immature germ cells. Here, mother and the two daughter cells have got the same number of chromosomes as well as the genes. So there is no shuffling or anything. Whatever the parent cell is having, the same genes are transferred to the daughter cells. Hence, it is a homotypical or equating division. So time taken for this is about 1 to maybe could be sometimes 2.5 hours also. 1 to 2.5 hours is the time taken. It is a symmetrical division. So here what happens, most of the cells of the body, they have the symmetrical division here. So both the daughter cells are equal in size and they have the same number of chromosomes, genes, everything is same. Now, one more type of mitosis is there, that is asymmetrical one. Here, what happens is one daughter cell is more differentiated than the other daughter cell. Clear? So, this is the basis of the stem cells. You would have heard no? Stem cells. They are destined to form many kind of cells, right, in the body. Like you have the stem cell in the stomach, okay, epithelium of the stomach. These stem cells, they can differentiate into any type. Here, there are so many types of cells in the fundus of the stomach. So they may differentiate into any type. During that division, what happens is one daughter cell differentiates more than the second daughter cell here, and that is how you see that stem cells are differentiation is called asymmetrical mitosis. So steps of mitosis, you know them, the prophase, metaphase, anaphase, and the telophase. So this is the cycle showing the mitosis. I'm not sure whether it is very much clear. Just pull down this slide here a little bit. Okay. So now I think it's visible, right? So in the mitosis or the somatic cell division, you have the prophase. Mute yourself, all of you. It disturbs the class. Then the metaphase, anaphase, telophase. So these all, they belong to karyokinesis, right? It is the division of the nucleus that is called karyokinesis, that is followed by cytokinesis. So what exactly happens in the prophase? So here is the cell depicting the cell membrane and then the nucleus with a well-defined nuclear membrane, right? So here, first thing is you should know is the nuclear membrane is still intact during the prophase. Here we see that the chromosomes, usually what will be the state of the chromosomes in a normal cell? Are they visible in the nucleus? No, they are not visible. Okay, they will not be visible. They are just strands will be there within the nucleus. So what happens in the initial phase of prophase is there is condensation of the chromosomes. Clear? So the chromosomes get condensed and they are held in the middle of the chromosomes with the two chromatids are there here, right? So they differentiate themselves, like the chromosomes, they condense and form the two chromatids are formed with the central centromere holding them. Here, this is the centromere which holds the two chromatids. What holds them is the kinetochore. This kinetochore is again a form of your micro, it's an envelope around that centromere. Clear? Now what happens in the cytoplasm at this stage? We see that the centrioles, they start moving apart. The two centrioles which are seen here, they start moving apart and initial formation of the spindles start here. Spindle formation, they just start appearing. Now what happens during pro-metaphase or just before the metaphase? So here we see that the nuclear membrane and the nucleolus, which is seen here, they have disappeared, right? And you see both the uh, centrioles are at the two opposite poles, and the chromosomes, which are condensed and have formed this pattern, they are just entangled in the microtubules. They are entangled there. Here in the microtubules. They are not arranged in any specific pattern here at the pro-metaphase. Then we'll move on to the metaphase. So here what is happening is the chromosomes arrange themselves at the equatorial plane. This is the equatorial plane of the cell with the centrioles at the two poles, and you see that definite pattern of microtubules are extending from one pole to the other, holding the centro centrosomes. Clear? So they will be holding from here up to the centrioles. So these are the specific microtubules which are meant for holding these centrosomes with the chromatids in the equatorial plane. Then during anaphase, what happens is there is shortening of these microtubules. Clear? There is shortening of the microtubules, and the centromeres, they help in, they split here. Clear? They split, and what happens is the chromatids are being pulled towards the opposite poles. Right? So each chromatid will be receiving one long arm and one short arm. If you know the structure of a chromosome, it will have one long arm and one short arm held together by a centromere, right? So each side you will get one long arm, one short arm, like that it will be, right? So equal distribution of the chromosomes occur here. There is, oh, why does it pull apart? That also you should know here. Because of the shortening of the microtubules. The microtubules shorten. When they shorten, they start pulling these centromeres towards their poles, right? So here the chromatids get separated, as you see here. Then begins the telophase. So in the telophase, you see that there will be constriction which has appeared in the cell membrane. We can see here constriction has appeared in the cell membrane. Then you see the polar microtubules are present, right? And the nuclear envelope, this is a stage of reorganization. Here, telophase is a stage of reorganization. So what you see is the nuclear membrane starts appearing here, reappearing around these polar centrioles and the chromatids, right? So here we see reappearance of the nuclear membrane, and constriction deepens here. What force is applied at the microtubules here? It is the centrifugal force which has been applied here. Then begins the cytokinesis. Once the nuclear membrane has formed itself, we see that at the mid body there is a full constriction which has appeared here, and only in the at the middle of this constriction we see that it remains as a mid body here. A contractile ring is created. It cleaves the cells into two equal halves. A well-formed nuclear membrane is there with the chromatin material inside. The chromatids, they rearrange themselves. So once the cell division, that means cytokinesis has appeared, what happens to these chromosomes? They they form their original format. That is, they are not condensed now. They become linear format will be there, and you will not be able to identify the chromosomes once they divide. So this is the end of the cell division. Now, there are certain things which you need to understand in these phases. So during the metaphase, what happens is the chromosomes are well arranged at the equatorial plane, right? In the metaphase at the equatorial plane. And you'll be able to identify these chromosomes if you visualize under the microscope. Here, during other phases, these chromosomes are not well visualized. Now, why do you want to visualize these chromosomes, which are paired, having long arm, short arm, right? That is the question. So it forms the basis of the karyotyping. Okay, karyotyping is you're studying the chromosomal pattern, number of chromosomes, how they are arranged, and whether anything which is abnormal is there in the chromosomes. You know that 23 pairs are there, right? In every individual, 23 pairs of chromosomes are there. But you also may be knowing at this stage that there are some anomalies in the body which may occur as congenital anomalies. Which are related to chromosomes, number of chromosomes, or could be structural anomalies in the chromosomes. If you want to know the number of chromosomes, then you can do this procedure, that is the karyotyping. So if you want to visualize the chromosomes under microscope and see how many are there and whether any abnormality in the number is there, then you need to do the karyotyping. You want to see chromosomes, right? That is why you need to see them during the metaphase. There is a drug called as colchicine. Okay, colchicine. This drug arrests the cell in metaphase. At the metaphase stage, the cell division is arrested. Clear? And we can easily see these chromosomes arranged. And you can stain these chromosomes and visualize under the microscope. Clear? That is one applied aspect which has a clinical relevance here. Then second is, sometimes in many diseases, there could be extra number of chromosomes may be there, or one chromosome is less in that individual, or it could be one, could be more. Clear? So why that occurs? Because there is abnormal function of the spindle apparatus. So when there is abnormal function of spindle apparatus, what happens is one chromosome fails to migrate to the opposite pole, right? So here we can see here they are migrating to the opposite poles. So if this part, if the microtubules at this part, if they are having, if it is not functioning properly, what happens to this chromatid? It will be pulled to the other side only. So what will happen to number of chromosomes there? Number of chromosomes in one cell will be more and in the other cell will be less. Here, so that leads to many abnormalities. So I will not point out right now the diseases here. It is called as the non-disjunction. Clear? So if there is abnormal function of spindle apparatus, that is called non-disjunction. It could serve as one of the basis for extra number of chromosomes, or maybe less number of chromosomes in one daughter cell than the other. Then second is one entity called as isochromosome is there. So here what do we see is, usually what happens to these chromatids, they longitudinally, it will be split horizontally like here, like this. Clear? It will be split horizontally. So what happens to daughter cells then? If any of the chromosome is split like that, one daughter cell will be receiving short arm, one will be receiving the long arms. So that is called as isochromosome. Clear? Isochromosome. So that also has got clinical relevance. So this is about the mitosis. Now I've got just one small video for you. I'll just play that. So share ಆಗ್ತದೆ. Okay.
Mitosis. In this module, you will learn about mitosis. Mitosis is a process of cell division that makes possible regeneration of body parts. To elaborate, mitosis is the separation of nuclear chromosomes into two identical daughter nuclei. It is divided into four key phases: prophase, metaphase, anaphase, and telophase. An additional phase called cytokinesis occurs after the complete nuclear division. Before a dividing cell enters mitosis, it undergoes a period of growth and rest. This period is called interphase. In interphase, the genetic material of a cell increases. Prophase. During prophase, the chromatin condenses and becomes short and thick to form chromosomes. Each chromosome has two sister chromatids joined together at the centromere. The nucleolus disappears and the nuclear membrane starts to break down. The centrosomes, each with a pair of centrioles, move apart to the opposite poles, forming a spindle between them. Metaphase. During metaphase, microtubules of the spindle interact with chromosomes. This causes the chromosomes to move and align along the middle of the cell or the equatorial plate. Sister chromatids are held at the centromere, confirming the presence of protein structures called kinetochore at the surface. The kinetochore holds the chromosomes and attaches them to the spindle. Anaphase. During anaphase, the sister chromatids separate at the kinetochores, and the daughter chromosomes, along with the divided centromere, move towards the opposite poles as the microtubule shortens. The poles of the spindle also move apart and help to separate the chromosomes. As the chromosomes separate completely into two sets, one at each pole, anaphase is considered complete. Telophase. During telophase, the daughter chromosomes arrive at the pole and the chromatin starts to decoil. Also, the nuclei reform along with nucleoli. This triggers the reformation of the nuclear membrane. Cytokinesis. Cytokinesis is the partition of the cytoplasm. In animals, a cleavage furrow appears in the plasma membrane, which deepens and divides the cytoplasm into two daughter cells, each containing a nucleus. In plants, new cell wall formation.
So you have watched that. Now we'll move on to next. That is the meiosis. I think the upper part of the slide is not visible. We'll move on to meiosis. So what is meiosis? It yields haploid cells. So what difference is there? In mitosis, you see that from the parent cell, you are getting same number of chromosomes are transferred to the daughter cells, whereas in the meiosis, it yields haploid cells from a diploid parent cell. It is called heterotypical division. It occurs in sex cells during maturation of gametes. There are two phases here, first and the second meiotic division. Now let us see about uh, the first meiotic division. Again, here both the meiotic divisions have got all the phases: prophase, metaphase, anaphase, and the telophase. So let us start with the prophase 1. So which is most important here. We see that the chromosomes, they start condensing within the nuclear membrane, and you see that they start aligning themselves with the sister chromatids. Clear? Now, the important part here is in this occurs in the germ cells, right? So here in the germ cells, we see that there is one chromosome which is derived from the uh, paternal part, and the other one is from the maternal side, right? So they align themselves, and what happens is you are seeing there is formation of synaptotenimal complex, right? So they are spiraled here. The DNA materials is spiraled. The chromatin is spiraled here, and you see that there is formation of tetrad. What happens is the each chromosome will be having a short arm, long arm, right? Now this is arranged in such a way that the chromatids will split, and in the center they are held by the centromere, right? So what happens in each, when you are seeing in each cell, we are seeing the formation of a tetrad here. Clear? So these are the subdivisions of the prophase 1: leptotene, zygotene, pachytene. Now in diplotene, what happens is these, they arrange themselves in a tetrad formation during pachytene. During diplotene, we see that the endonuclease enzyme will help in crossing over of these sister, these are the sister chromatids, right? So here there is crossing over. That means part of the chromatid will move to the opposite side. So we are seeing there is a shuffling, reshuffling, I can call it, instead of shuffling, reshuffling of the genetic material, which is a very important step which is occurring in the prophase, right? So this is very important because the offsprings which are born, they are not exactly like the parent cell. That means you do not exactly resemble your parents, neither your sibling, your maybe your sister, brother. They don't belong to you as you exactly 100%. That is because of reshuffling of the genetic material which occurs during the crossing over here at the diplotene stage. Then during the diakinesis, you see that there will be disappearance of the nuclear membrane. The centrioles, they start moving to the opposite poles, right? So this prophase 1 is very important stage in the meiosis because there is formation of, however, in both the mitosis as well as meiosis, there will be replication of DNA that will occur during the interphase, right? After that, when it enters the prophase, the formation of tetrad, the crossing over, they occur in the meiosis to maintain the uh, genetic diversity. Clear? Hereditary diversity should be there. To maintain that, you are seeing crossing over. Then what happens is in the metaphase 1, you are seeing that it is somewhat similar to your mitotic phase, but one important difference is there. So the centrioles, they move apart. Then you see that the chromosomes, they arrange themselves with their uh, crossed over gene, I can tell, right, along the equatorial plane. So what happens here? This is the prophase, right? In the metaphase, they have arranged themselves at the equatorial plane. Then what is happening is the centromeres of this tetrad, they don't divide here. What will happen is the chromosomes, they just move apart here. Here,